Bismillahirrahmanirrahim. Welcome again, guys. Welcome back to another video lecture by Kami microbiologist. Today in this video lecture we are going to talk about another biochemical test, that is a phenylalanine agar test. Okay, so we already talked almost 26 videos related to biochemical test. There will be also a playlist if you want to check all the video, and if you want to see all the video, there will be a playlist. You can go back to my channel and watch the playlist related to biochemical test. It will contain all the biochemical tests that is very important in the microbiology. So this is our 27th video lecture, and in this video we are going to talk about 26th biochemical test, that is related to phenylalanine agar test. So phenylalanine agar test is also known as phenylalanine diminase test, or we can say diminase medium. Actually this is called diminase medium because in this medium or in this test the diminase enzyme was detected, and this diminase enzyme was actually detected in the protease species by a scientist named Hendrickson in 1950. Okay, after that another scientist his name was Ewing E.H., and in 1957 he actually a little bit changed the media formulation, each and everything, and then he due to this way he detected the bacterial ability that can dominate the phenylalanine oxidatively. After that another scientist, his name was Edry, in 1971 he actually prepared a disc that is used as a rapid test, in case of phenylalanine agar test, and he just check the ability of the bacteria that can produce urease and phenylalanine diminase enzyme. Okay, now the fifth point says that we better know that phenyl pyruvic acid, it is actually formed during this reaction, and when it is formed during this reaction, then it is also called phenyl pyruvic acid test as well. Okay, so this is a little bit introduction related to phenylalanine agar test. Next is objective or uses. This type of the test is specially used to just check the ability of the bacteria that can produce phenylalanine diminase enzyme. Okay, and this test is also used to differentiate the protease species, Morganella species, Providentia species, among other Enterobacteriaceae like E. coli, Klebsiella. Okay, now then we have the requirements. So if we want to perform that test, what we need phenylalanine agar medium, we need autoclave, incubator, personal protective equipment. In personal protective equipment we need goggles, gloves, cap, overall. Okay, all these things that we are really that's really important. Next we need petri dishes, conical flask, and other lab material. Okay, so other lab material means like glasswares, spirit lamp, okay, LFH, all these things we also need. Next we need ferric chloride solution. So ferric chloride solution is prepared that we need 10 gram of the ferric chloride powder. We will mix it into 100 ml of distilled water. After that we will mix it into 2 ml of HCl. Now we will mix all these things, and then we need what we also need urea PDA disc and 1 NHCl. So these are the main things that we need in case of the rapid test and in case of the simple test. So in the next slide I will show you that we performed two methods while performing that test. Now as we talk that we need the media, so what is the composition of the media? So the composition of the media is that it contains yeast extract 3 gram, DL phenylalanine 2 gram, sodium chloride 5 gram, disodium hydrogen phosphate 1 gram, agar 15 gram. Okay, so you will mix all these ingredients in 1 liter, then you will get the phenylalanine agar medium. The test organism, okay, or if you want to check the quality of this media, so you have to be you have to positive control and negative control. In case of the positive control, you have known Proteus mirabilis species as well as E. coli, that is acting as a negative. Okay, so these two things you have two things, these two bacteria. And once you have these two bacteria, so then you will also perform your test, and with the positive and negative control you will check the quality of the media. And due to this way, if you get the positive result and if you get the negative result with these two bacteria, it means that your media is right and the quality is okay. Then the test as you perform, each and everything is fine. So this is how the quality control is very important in this case. Next we need principle. Okay, now what's the principle says of this test? So the principle says that we better know that in this case it contains the phenylalanine. So the phenylalanine is actually the substrate, okay, for the enzyme that is the deaminase enzyme. Okay, so when both substrate and enzyme are react with each other, what will happen? It will deaminate. Okay, so it will deaminate, and after that what we get the phenylpyruvic acid. This is the reaction. Next point says that yeast extract that is available in this medium, it is important to just support the growth of the organism. Next we need sodium chloride that maintain the osmotic equilibrium. And next we have okay, so these are the main thing that is present, and that is present into the medium, and that's the most important working of this ingredient. Next we have the reaction, that what's going on. Let's suppose you have streaked the bacteria into the medium, then after that what will happen? The bacteria will produce enzyme, that is the phenylalanine deaminase enzyme, that will remove the amine from the phenylalanine. And then as a reaction, what it will produce? It will produce ammonia as well as phenylpyruvic acid. Okay, and then phenylpyruvic acid is detected by adding a few drops of the 10% ferric chloride, which act as a chelating agent. After that we get the green color complex that is formed between these two compounds, indicating a positive result. Okay, now the second the last point of this principle says that if let's suppose you get the medium color straw in color, so the organism is negative for phenylalanine deaminase production. You have media preparation. So how you will prepare the media? So the media preparation is very simple. You need the media bottle that is available commercially. If you don't have the bottle, so you have to be the ingredient as we talked in the previous slides. Okay, so you will mix all the ingredient in 1 liter. If you have the powder, so you will mix the powder into 1 liter. After that you will heat to mix it completely. Heat once it is heated, so what will happen? It will be completely dissolved. After that you will autoclave it. Autoclaving should be 121 degrees Celsius for 15 minutes, and the pressure would be 15 psi. After the autoclaving each and everything, you will cool down the media. The temperature will be reached 45 to 50 centigrade, and then you will mix and pour the media into sterilized test tube. 5 ml you need the media, and you will put it into sterilized test tube. Then what will you do? You will solidify the test tube to the slant position. Okay, so this is how you will prepare the media. Next, the method of the test. So there will be the two method of the test, that is the agar slant method, and the next one is the rapid method. So agar slant method procedure, what will you do? First of all, you have to fresh your bacteria, that bacteria which you want to identify. Okay, once it is fresh, so then you have to take heavy inoculum, okay, heavy bacterial colony, and then you will streak the heavy bacterial colony into the slant portion and as well as the butt portion, etc. Once it is streaked, so then what will you do? You will incubate it for 6 hours. Why? Because you take heavy inoculum. After 6 hours it will give you the result. Okay, and if you take a light inoculum, so it will take 24 or 18 hours. Okay, now after incubation, once you get the result, so what will you do? You have to add 4 to 5 drops of the acid 10% ferric chloride solution directly over to the slant, and then you will gently roll the slant, due to which it will reach over the all slant surface. Now once it's done, so then you have to wait 5 minutes. Within the 5 minutes you will check the color from green color, okay, and or the development of the green color or dark color, it will indicate what? It will indicate the positive reaction or positive result. Repeat method. The repeat method is just like that. You just take a calcium tube or a test tube, and then you will add 25 microliter or 0.2 ml or 0.25 ml of normal saline, and will make a heavy suspension of the bacteria, test bacteria, okay. And then what will you do? You will place and or we can say add PDA disc, a urea PDA disc over to the this suspension. After that, what will you do? You have to incubate the tube for 2 hours at 37 degrees Celsius. Now after 2 hours, you will check the pink color development, that will indicate positive urease test. And if there is no color, so it will be negative color. After that, you will add 1 NaCl 10% ferric chloride solution, and you will gently shake it and observe the development of the green color. This is how you will perform the repeat method. This is the result, as you can see. So the development of the light green to dark green over the slant, we can say like that, within 1 to 5 minutes, it will indicate the positive. As you can see this second picture, this is the positive result. And if you see the absence of the green color development indicated a negative reaction. So there is no green color, dark green color, so it means this is the negative reaction. Now this is the result, and let's see which type of the bacteria give us positive result and which type of the bacteria will give us negative reaction. So the positive bacteria, PDA positive bacteria, is the Proteus species, Morganella species, Providentia species. These are the positive bacteria, PDA test positive bacteria. PDA test negative bacteria are E. coli, Klebsiella, and also some other Enterobacteriaceae as well. Now the most important thing, that is the precautions. What will you do? So the precaution is that you have to take heavy inoculum, okay. Heavy inoculum is really important. Next, you have to store the ferric chloride solution into the dark bottle, into the dark space away from the light, otherwise there will be a reaction and the ferric chloride will be, not working. Third point says that you have to check the urea PDA disc for discoloration before use. You have to check the color. If the color is changed, then it is not important that you will use it, okay. So these are the main precautions that you have to adopt it while performing that test. There is main thing that I didn't write, that is the limitation. Limitation means that you have to perform other biochemical tests, because when you want to know the proteases, so the Proteus, Morganella and Providentia have the same characteristics in this test. So it means that for one particular bacteria, this one test is not important. You have to perform other biochemical tests to confirm the bacteria 100%. Okay, so this is all about the PDA, or we can say phenylalanine agar test. I hope this video is helpful for you. Thank you so much for watching.